Welcome to PK's classes. Today we are going to study on tryptophan operon. Tryptophan operon is also known as TRP operon. So TRP here stands for tryptophan. So we know tryptophan is an amino acid, and this tryptophan is synthesized in the bacteria Escherichia coli, E. coli. So the E. coli chromosome has the gene for synthesis of tryptophan, and as you know, you can go to our class on operon, you can see that, and we have discussed already the lac operon. So today we are going to discuss about the tryptophan operon. As you know, bacteria is a prokaryote, a single cell unicellular without any nucleus, and it has chromosome. So this chromosome contains this TRP operon, and the regulatory gene is known as TRPR, which is away from this operon, whereas in lac operon it was with the operon. Here it is away from the operon. So you can see what is the structure of the TRP operon. So this is the TRPR gene which produces the TRP repressor. This regulates the tryptophan synthesis, and the TRP operon first there is promoter to which RNA polymerase binds, operator site to which the TRP repressor binds. Then there is a leader sequence, and there are 4 sequences: sequence 1, sequence 2, sequence 3 and sequence 4 in this leader sequence. Then there are the structural genes, and here in this TRP operon there are 5 genes: TRPE, TRPD, TRPC, TRPB, TRPA. So these 5 genes together produce the enzymes required for tryptophan synthesis. So we call them as the structural gene, and this is the TRP operon. So this is the structure of the TRP operon, consisting of promoter, operator, leader sequence and the structural genes, and finally there is a terminator. So to the promoter RNA polymerase binds, and to the operator this TRP repressor protein binds. But the TRP repressor protein which is produced from the TRPR gene is usually inactive, and this becomes active in presence of tryptophan. So tryptophan activates this TRP repressor, and in this way this TRP repressor protein regulates the tryptophan synthesis. So accordingly the regulation of TRP operon occurs in 2 ways. One is negative co-repression and the second is attenuation. Negative co-repression that is through tryptophan, and attenuation occurs through the leader sequence. So this is the leader sequence which regulates the tryptophan synthesis. So that is we call as attenuation. And the other one, this repressor protein regulates the tryptophan synthesis. So this is through TRP repressor protein. And this tryptophan operon is a repressive system, unlike the lac operon which is an inducible system. So why we call it as repressive system? Because normally the gene is on. This repressor protein which is produced from the TRPR gene is unable to bind to the operator. So RNA polymerase once binds to the promoter, it can move and can cause the transcription of the genes, and then produces the enzymes for the tryptophan synthesis. So RNA polymerase binds to the promoter region and it moves. When it moves, it causes the transcription of all these genes or DNA, and accordingly produces the enzymes for tryptophan synthesis. So this TRP repressor protein, if it, suppose this is the structure, it cannot bind to the operator, and once it is activated, the structure, suppose say, becomes like this, and it binds to the operator and inhibits the action of RNA polymerase. So what happens? Normally the gene is on because this TRP repressor protein is not able to bind to the operator. So out of these two systems, the regulation by two mechanisms, negative co-repression and attenuation. Today we are going to discuss about the negative co-repression. In the next class we will discuss about the attenuation. So what happens? Normally the gene is on and the gene transcription goes on because the repressor protein which is produced from the TRPR gene is not able to bind to the operator, so it is not blocking the RNA polymerase activity. So RNA polymerase activity goes on, gene transcription occurs, and enzymes for tryptophan synthesis are produced. But when the tryptophan is synthesized, tryptophan now binds to this repressor, makes it active, and it blocks the operator. So RNA polymerase now cannot move, and there will be no transcription of the structural genes, no enzymes produced, so decreased tryptophan synthesis. So, so when there is no tryptophan or low tryptophan, gene transcription occurs, enzymes for tryptophan synthesis occurs. So tryptophan synthesis usually occurs, and when there will be tryptophan, that tryptophan will activate the repressor protein, which will block the operator, and there will be no transcription, no enzymes. And so let us see how this tryptophan amino acid is synthesized. Tryptophan is synthesized from corepressor. Tryptophan is synthesized from corepressor by the action of anthanylate synthase, and corepressor produces anthanylate by the enzyme anthanylate synthase, and here we require glutamate, which is converted to glutamate pyruvate. Then anthanylate is converted to phosphoribosyl anthanylate. In presence of anthanylate, phosphoribosyl transferase. Then phosphoribosyl anthanylate is converted to CPDP, carboxyphenylamino deoxyribulose 5-phosphate, in presence of phosphoribosyl anthanylate isomerase. Then by IGP synthase, indole 3-glycerol phosphate synthase, indole 3-glycerol phosphate is produced, and then by the action of tryptophan synthase, tryptophan is produced. So indole 3-glycerol phosphate first releases 3-phosphoglycerolaldehyde to produce indole, which in presence of serine and tryptophan synthase produces tryptophan. So for tryptophan synthesis we require 3 synthase enzymes. One is anthanylate synthase, and 2 is IGP synthase, and 3 is tryptophan synthase, and the other 2 enzymes are isomerase and transferase. So these 3 enzymes are produced from these structural genes. As we know, gene on transcription produces messenger RNA, then messenger RNA on translation produces protein. So TRPE and TRPD gene produces this enzyme anthanylate synthase. Enzymes are nothing but proteins. So anthanylate synthase protein is produced from TRPE and D gene. TRPC gene produces indole glycerol phosphate synthase or IGP synthase, and TRPB and A produce the enzyme tryptophan synthase. So these 3 enzymes are produced from the genes. So the genes first undergo transcription to produce their specific messenger RNA, and messenger RNA then on translation produces these proteins or enzymes. So that leads to synthesis of tryptophan from corepressor to tryptophan. So the regulation of TRP operon, the two mechanisms, negative co-repression and attenuation. Today we have discussed the co-repression, where normally the gene is on, and why the gene is on? Because the repressor protein which is produced from the TRPR gene, the regulatory gene, is not capable of binding to the operator, so RNA polymerase activity is not blocked and there will be gene transcription. And the other one, when there is enough tryptophan, that will bind to the TRP repressor protein and makes it active so that it will block the operator, and the RNA polymerase which binds to the promoter site now cannot move on, there will be no transcription. So you can see that. So negative co-repression, low tryptophan, no binding with TRP repressor, inactive TRP repressor, so it fails to bind to the operator, so there will be no inhibition of RNA polymerase activity, gene transcription occurs, messenger RNA is produced, enzymes for tryptophan synthesis occurs, tryptophan increases, high tryptophan, then we will discuss high tryptophan. So this is one. Okay. So when there is low tryptophan, there will be increase in tryptophan. So when there is high tryptophan, what happens? High tryptophan binds to the TRP repressor, makes it active, it blocks the operator, so there will be no RNA polymerase activity and it will inhibit the transcription of TRP genes and there will be decrease in tryptophan synthesis. So this is all about the regulation of tryptophan synthesis by the TRP operon through negative co-repression. In the next class, we will study in the next week about the attenuation. Thank you.